And so now, without further ado, I would like to introduce you to Mandana Arbab, our first and keynote speaker. She's the Lodish Family Assistant Professor at the Rosamund Stone Zander and Hans-Jörg Wyss Translational Neuroscience Center at Children's Hospital and Harvard Medical School in the Department of Neurology. But Mandana is no stranger to the Broad. She was actually here in David Liu's lab for some time prior to this academic position. And so it's really a distinct and special pleasure to welcome her back to give us her keynote lecture. Thank you, Mandana.
Thank you very much for that fantastic introduction. I want to jump right in with a graph basically showing the distribution of genetic variants that are causal to human disease. As you can see here, the overwhelming majority of the pie is made up of point mutations. And the reason that point mutations are so heavily contributing to genetic disease is the fact that it's the most common form of DNA damage that our cells undergo, literally hundreds to thousands of times a day in every single cell of our body. And this happens due to chemical reactions like oxidation, deamination, and other spontaneous events. And typically, these are restored by the cell correctly, but occasionally they're not. And that can lead either to what we see as heritable or spontaneous or somatic diseases. And there are enzymes in our own cells that also contribute to these sorts of mutations. And in a feat of very brilliant engineering that was done by Alexis Comore and Nicole Gaudelli in David Liu's lab, who the place where I also worked previously in my postdoc right here at the Broad, we are now able to basically harness these enzymes to our advantage to revert exactly these sorts of disease-causing single nucleotide variations with a technology called base editing. And that is a technology that our lab primarily uses along with other precision gene editing tools to try to tackle rare diseases. And that's why I wanted to highlight this for you. And I know we have quite a mixed audience here of, expert developers of these technologies and users, but also non-scientists, patients who might be less familiar with these technologies. So before I talk about all the things that we can do with these tools, I just wanted to give a really brief overview of this technology that now at this point has actually very recently, very successfully been used not only in ex vivo applications, but also even the very first in vivo therapy in young baby KJ, as I'm sure most of you are well aware of. So base editing is this technology that uses part of the CRISPR system to do precise chemistry in the DNA of living cells. The protein that's shown here, Cas9, pairs with a guide RNA that's shown in purple to localize the editor to a user-specified location in the genome. And there, the appended deaminase enzyme here and the A-base editor is able to do a chemical conversion on a single strand of DNA. And so what happens in this case, for example, is when you have a TA base pair, the enzyme deaminates the A into an inosine, which is an unnatural nucleotide in DNA that is read as a G. And this results in a GT mismatch that then, through other clever tricks of engineering, we are able to push to predominantly result in a successful CG base pair over here. So this potential theoretically holds the technology to revert over half of the disease-causing SNVs that I showed you in the previous slide. And in a key study that we led, we demonstrated the therapeutic potential of this technology for a disease called spinal muscular atrophy. SMA, as it's abbreviated, is the leading genetic cause of infant death globally. It is a motor neuron disease, and that causes progressive muscle weakness, starting proximally, traveling distally, and if left untreated, patients typically succumb to respiratory distress. And it's caused by the loss of this gene called SMN1. And patients always retain copies of a very similar gene, SMN2. So while SMN1 is predominantly responsible for the production of SMN protein, SMN2 genes can also produce some amount of that. And depending on the number of copies of SMN2 that patients carry, it determines their disease severity. And in the most common form of the disease, type 1, patients have onset of symptoms almost immediately after birth, and if untreated, will typically succumb to the disease in about 18 months. whereas patients who have higher copy numbers, which is more rare, maybe 4 or 5 copies, might not present with symptoms until adulthood with proximal hypotonia. So I wanted to highlight this because SMA is also really interesting in general in the rare disease world, because it's really been a trailblazer in the development of gene-based therapeutics for a number of key reasons that I thought would maybe be interesting to highlight here, because some of these are immutable characteristics of the disease itself, but then there are other key elements that made SMA a really good disease target to go after and really help to make effective treatments for this disorder a reality. So for one thing, in the mutable category, there is the presence of a targetable gene in all patients that's always the same sequence. So as opposed to typically where you have a different mutation maybe for lots of different patients, there's always this one backup gene that looks exactly the same. So from a genetics perspective, that's excellent because you know exactly the sequence that you're trying to target. Then we also know, because of how the copy number determines disease severity in patients, that the disease is in fact dose responsive to SMN protein, and any more of it is very helpful. But at the same time, it's thankfully not a very dosage sensitive gene. Some diseases you can't really overexpress it too much either, but we know from now the gene therapy that in patients there doesn't seem to be necessarily that much overtoxicity related to overexpressing it. But also another key factor is that the protein that we're rescuing is already a self-antigen. So, in some cases, patients will have a mutant form of the protein. That's all their body's ever seen. And so while correcting it is obviously the goal, if their body's not familiar with the normal full-length version of that protein, you can actually even have an immune response against that. So it makes it quite complicated. SMA definitely benefited from these. But other, which I consider to be more actionable elements of this as a disease target, is that the genetics of this disease were extremely well understood. The gene had been identified at this point, I think maybe over 3 decades ago. But aside from just knowing the gene, there was a lot of extremely deep biology done on this gene to understand the regulation of both the transcription and the protein product. And this was done by several groups, including Adrian Krainer's group, Arthur Burgess, Gideon Dreyfus, and several others. And because we understood so well the regulation of this gene, we were then able to exploit that for a number of the drugs that are now on the market. So that I think has been really key. At the same time, having a clear picture about how the genetics also relates to the disease also helps a lot with how we can make a diagnosis very early, ideally even before the onset of symptoms, which is often quite desirable for a lot of these diseases. And then pairing that with very strong natural history data allows for effective clinical trials to be designed. And this is where the roles of patient advocacy groups is huge. And the groups shown here played a major role in being able to make these a reality. And then finally, what I think is also important is there were very well established genetic disease models of this disease for SMA that were present. And they were really key in translating the current drugs that are on the market to getting them to the clinic. These were genes that had the human copy of SMN2 knocked in, so you have something to target there. It also really faithfully recapitulated the disease phenotypes. At the same time, now, that might not necessarily be a requirement anymore to get these drugs approved, which I think is a very key and important point, because we're very lucky that you can even model this disease well in mice. That's not something that is true for most diseases. And so if that is holding you back, we're never going to be able to realize this future that we're hoping for of accelerating the translation of these new therapeutics. And as we saw, we've now repeatedly seen, with the development of personalized ASO drugs, but also, for example, in the case of baby KJ, where it was a gene editing drug, a model that's maybe as simple as, a knock-in, not even at the endogenous real locus of the sequence, just to be able to demonstrate that you can then do the editing there through the, plausible mechanism pathway might be sufficient, as long as you can pair that with a really strong scientific basis as to why doing such a corrective edit, for example, should work. So I think that, being able to move away from having to make very intricate mouse models, or even more simple in vitro models, like having individual iPSC lines and organoids that you can make that can be really cumbersome, will really hopefully help this field move forward. Anyway, I wanted to sum these up because, it's still kind of early days in translating gene editing technologies, but for that reason, we want to be very careful in ascertaining what targets to go after, and these are certainly the key factors that go into that calculus. So in the case of SMA, as I mentioned, there are several gene therapies that are already, or not gene therapies, gene-based therapies that are on the market, including one gene therapy, Zolgensma, as well as an ASO drug and a small molecule, which is first-in-class small molecule splicing modifier. They're really absolutely game-changing, very effective. A couple of limitations of these drugs as it still stands is that the ones that act on RNA are only transiently effective. You need to have repeated oral dosing or an intrathecal injection several times a year to maintain efficacy. And in all of the cases, none of the drugs actually upregulate the protein levels to the level that we want. So it's typically downregulated by about 6-fold in the spinal cord in SMA patients, and these drugs only can upregulate by about 1.5 to 2-fold, and they're also not able to facilitate sort of the responsive, fluctuations that your bodies have of SMN levels throughout your lifetime. So having a different approach that allows for a one-time treatment that will guaranteed to be permanent, that does allow for endogenous expression and native regulation would be an advance over the existing therapies. And so we developed an adenine base editor approach to do this exact thing. So the SMN1, which is the gene that patients lack, differs from SMN2, this insufficient gene, by just a single nucleotide change that results in a splicing defect. And so what we're able to do is we're able to restore this TA to CG and basically revert to the normal SMN gene that we all have. And we were able to demonstrate that by delivery using the vector AAV9, which is the same AAV vector that's used for delivery of Zolgensma, the gene therapy that's on the market, we're able to effectively target spinal motor neurons to the same degree as you can with Zolgensma. And in neurons that receive the editors, as shown here, 90% of them have conversion of the SMN2 gene to the SMN1 gene. And what that results in, if we look at the number of motor units, which are typically lost, as is seen here in untreated animals compared to healthy controls, is when we treat with our editor, we can restore this number significantly. And what I think is probably more telling is to actually see a video of how they act. So what we're doing here is called a righting reflex assay. Mice don't like being on their back, so when you tip them over, they try to scramble and right themselves. And you can see here at 14 days old, an SMA mouse is poorly capable of doing so. And once we treat the animals with our editor, you can see that they're a lot more spry and able to right themselves. And I think what is a more significant readout than that even is the survival. So, we're intending to take this to clinical trial. A trial at this point for SMA patients would, of course, involve treating patients with the standard of care. What we have demonstrated is compatibility as well of our approach with an existing standard of care, the ASO drug. And what we can see here is that if you administer just a single very small dose of the ASO drug to SMA mice at the day of birth, you can bump up their median survival from 17 days in an untreated animal to 29 days in the ASO treated group. And if we pair that then with our base editor drug, we boost the median survival to 77 days. And as you can see here, here's an animal at 200 days old with one of its litter mates. It does suffer from some strain-specific effects of the SMA phenotype that humans don't experience. For example, it has lost its tail. We don't have a tail. But it also, as you can see, is mobile. It's walking around. It is not suffering from SMA. So our group is part of a government-funded effort called the Somatic Cell Gene Editing Consortium, together also with David Liu's group and the rare disease group at the Jackson Labs, aimed at accelerating the translation of gene editing therapeutics, and this SMA strategy is our leading strategy, and we're aiming to submit a pre-IND request later this spring. Another disease that I wanted to highlight is ALS. So SMA is kind of a very straightforward, there's one genetic mutation, if you can call it that, that you want to correct. But there are other more creative ways that you can use these same tools as well. So I wanted to highlight a couple other projects to that point, ALS being one of them. I'm sure you're all very well aware of ALS. It's another rare disease. There's 5,000 new cases diagnosed in the US every year. There are 7 approved drugs for it, but none of them really can alter the course of the disease, and patients still typically succumb due to respiratory distress. Now, ALS in 90% of cases is sporadic, which means there's no heritable component to the disease. And then in about 10% of ALS that is familial, there's about 40 different genes that are implicated in this that explain maybe 70% of it. But it's safe to say that the genetics of ALS are complex at best, and it's certainly not the only thing that's underlying the disease. So all of this at face value does not make it a great target for a gene-based therapeutic, of course. But nearly all patients, regardless of the cause of the disease, have a proteinopathy of this protein TDP-43. And this is a key splicing regulator, and as a result of it, there are several key genes that we now know are misspliced, and it is now thought that it is the loss of those genes' functions that is actually causal to the disease phenotype. So in one project, that we're working on, we're targeting one of these genes to restore that in ALS patients. And so the reasoning for this is that, as shown here, when you have a knockdown of TDP-43, we can see this gene STAthmin2 is exquisitely sensitive to the loss of TDP-43, even more so than the gene itself. And as collaborators of ours have shown, is that when you culture ALS neurons, or if you knock down TDP-43 in iPSC-derived neurons, and you do something called an axotomy, where you get rid of the axons, you have an impairment in their ability to regrow, which is something that neurons should natively be able to do. And if in that situation where you knock down with TDP-43 or you have an ALS neuron, if you complement with STAthmin alone, that is able to restore that axon regrowth. So what we did is we developed a base editing strategy to be able to correct this missplicing, and we have a number of strategies that all work quite efficiently. And importantly, if we then look at the splicing, if you see here wild type cells that have this normal splicing phenotype versus a mutant, we can significantly restore the full-length STAthmin products that are generated, and we have almost complete loss of the truncated form of STAthmin that is generated from here. And then in a final topic, I'd like to touch on another sort of more different way of applying these precision gene editing tools. We're going after trinucleotide repeat disorders as well, using base editing. This study was recently published in Nature Genetics, and I also wanted to highlight that this work was supported by the Friedreich's Ataxia Research Alliance here through the Broad, through the Friedreich's Ataxia Accelerator. And it's been really wonderful to work in that consortium, so very grateful for the opportunity to have been a part of that. So trinucleotide repeat disorders are a group of about 40 different disorders that are caused by somatic expansion of very small repeat sequences such as CAG, CTG, or GAA across various different genes. And the number of repeats at a given locus does vary in the population. However, once they reach over a certain threshold, they become unstable in some somatic cells. And so patients that have over a threshold length that differs by locus and by sequence will then have expansion of these sequences, as can be seen here. For example, CAG repeat size of 80 over time. You can see that there's a distribution where they sometimes also shorten, but mostly extend in length. And that ultimately leads to pathology. In many cases, these are neurological conditions that causes cell death. What is also known is that the length of the starting repeat is a very strong determinant of both the age of onset and the severity of the disease progression. So the longer your starting CAG repeat length, for example, as is shown here, the earlier your age of onset for the disease will be. What had been less appreciated until pretty recently is that the exact sequence of these repeats themselves are actually key drivers of the disease progression as well. So typically, Huntington's disease is, the first gene identified to be associated with a genetic disease, is often considered a polyglutamine disorder because it is caused by CAG expansion in the first exon that encodes glutamine. CAA also encodes glutamine. So in most people, the glutamine tract in exon 1 is CAG, 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 and then there's a penultimate CAA and then CAG again. In Huntington's patients, oftentimes that penultimate CAA glutamine is lost. And what careful studies have now shown, both through GWAS studies as well as pedigree studies, is that having even a single CAA interruption in that CAG repeat can delay the age of onset of Huntington's by up to 25 years, as has been found. So it's very profound. And it's now becoming more apparent that it's not actually the polyglutamine length that determines your age of onset, but more accurately, if you look at the stretch of the polyglutamine that is pure CAG, that is the more accurate determinant of your age of onset and disease progression. And the same is now being observed more for Friedreich's ataxia, which is a disease where it's not coding, it's in the first intron. There's a GAA expansion. And here, I have to note, these sequences are very difficult to sequence through because they're so repetitive. Our sequencers do not like that, and it's very hard to get accurate data on that. But now that we're realizing that in Friedreich's ataxia patients, similarly, when there is an interruption usually it's an extra G put into there, it can significantly delay the age of onset as well. So CAG to CAA and GAA to GGA or GAG are things that we can do with base editing. And so we developed base editing strategies to do just that, to sprinkle in base editing single nucleotide mutations in these repeat expansions. And so I'm showing you here, for example, is a patient fibroblast cell line from a Huntington's patient that has two alleles, one with 18 repeats long and one with 180 CAG repeats long. And we treated these fibroblasts with a cytosine base editor. And what we can see here is 5 days after treatment, this is what the distribution looks like. The untreated is behind here in blue, and in purple are the CBE treated ones. And here, 30 days after treatment, we can see that there is a significant difference in the distribution of repeat alleles of the long allele only in the CBE treated group versus the untreated group. And they are much shorter. They're actually even a little bit smaller than they were in the starting, but they have not undergone this expansion that is seen here in blue. And we've also demonstrated this in mice. So again, we've delivered here our editors to a Huntington's mouse model. This is one that has 110 CAG repeats. And it has expansion in certain cell types, including the cortex and the striatum, not the tail. And what we do is when we deliver this, we can see, if you look at the green bars, these are nuclei that are sorted that have cells that have been transduced. We can see that we get pretty good transduction up to at least 75%. We can only actually sequence a portion of it, so actually the real editing is a little bit higher than this. And this results then in a loss of this instability and expansion. So if you can see here the distribution of allele lengths in control here in gray, we can see that that hump here has been reduced in animals that have been treated with the editor, and you even have this group of contractions. And by this calculation called the instability index, we can look at the stability of these repeat alleles over time, and we can see, in particular, as time goes on, there's a great reduction in the instability of these alleles in treated animals. And the same we did for Friedreich's ataxia, where we looked at two different mouse models that had either 300 repeats at the FXN locus or 800, and after treatment with our adenine base editing strategy that installs A to G changes in the repeat tract, we see significant rescue of instability in the cortex of these animals. So with that, we're hoping that we'll be able to, as mentioned, through in part the SEG and other efforts, make these sorts of approaches a reality for therapeutics in the future. I want to thank all of our funding sources that make this work possible, again, including the Cure SMA Foundation, the Friedreich's ataxia Research Alliance, and all the teams who have worked with us on this work. Thank you.
Thank you. Beautiful talk. How do you safeguard? I'm not a gene editing specialist. How do you safeguard in off-target editing, especially in these repeat disorders, which I thought those repeats are pretty prevalent in the genome?
Yes, indeed they are. That, I think, would be sort of the major caveat of wanting to do that. So we can't, at this point in time, we don't have great technologies to safeguard against them. From, our interactions with the FDA on other projects, I think the major goal is mostly to sort of understand what they are, characterize them deeply, and monitor them. And so we've done extremely deep characterization on the off-targets of these strategies. And thankfully, while they are indeed plentiful because there are a lot of these, let's say, throughout the genome, for the most part, we don't think that they should necessarily have that big of a clinical impact because the mutations that we're making are synonymous changes, overwhelmingly. There are very few, like single digit, I think 1 or 2 sort of non-synonymous changes that we see as a result of this, in part because GAAs, for example, are almost all non-coding, and in the case of CAG repeats, they are more frequently coding, but the predominant outcome is CAA. So it is a difficulty. I think in a sort of broader sense, with the idea of wanting to translate gene editing therapeutics for in vivo applications that we're delivering by AAV, is the fact that, yes, there is no off switch. And so it's not just sort of the edits, the off-target edits that you might accumulate at that point where you're doing your important on-target editing, but it's the potential for accumulated off-target edits that happen over time. And we do see with our repeat targeting strategies that you have quite a lot of potential for that because we see an increase in editing at 24 weeks compared to 12 weeks. So I think the answer to that is that we're not yet there in translating these approaches to the clinic, but having an ability to have regulation of base editor expression, I think, will be the best way that we can have to safeguard against potential bad off-target effects.
Any more questions? I'll ask a quick question. First of all, it's beautiful to see stathmin. I told you earlier that I worked on it in graduate school, but actually it's a nodal mechanism, right? So it's very interesting because it can address multiple potential causes of ALS, so it's very interesting. I wanted to ask a technical question that has to do with exactly, these repeat disorders. You mentioned that sequencing is difficult in these areas. And so I'm just curious about both from the research perspective, but also from the patient identification perspective. If you're looking for people with specific repeats and it's hard to sequence in that area, do you see long-read sequencing as a solution? How do you see the deployment of that? Or what other solutions would you have for making sure that you pick the right patients and that you make the right edits, in the clinical setting ultimately?
Yeah, it's an excellent question. It's very complicated. We have, for these projects, used or tried to use pretty much about every form of sequencing that is available. Also non-sequencing, so sizing you can actually do a little bit more accurately than trying to get the exact sequence. In a clinical setting, even sizing can be quite complicated depending on the disease. For one thing, in Huntington's disease, you're talking about in the brain. So how are you even going to, extract those cells out to sequence them? You won't. So for the most part, you're going off of germline repeat length. For Friedreich's ataxia, it's additionally more complicated because these repeats can sometimes be like thousands long, which gets very challenging with any sequencing technology. So currently, none of the sequencing technologies are that good at faithfully reporting on what the exact sequence of these is, but I don't think that that's necessarily critical. There was a paper that David Lynch worked on that pretty recently came out that actually showed that even our sizing by PCR that we were doing has been wrong, and they found better ways of being able to do these PCRs. So I guess the answer to your question is if we got to a point of wanting to apply this in people, I think we would probably mainly go on sizing, and then knowing that these edits do take place, basically. But it is extremely challenging, and I hope anyone who's expert on sequencing technologies can work on this specific problem because it is a huge problem for us.
Yeah, we'll give it as a throwdown to the genomics platform. Throwdown to the genomics platform at the Broad to solve.
I really would appreciate that.
They know that I've bugged them about this before too. So thank you so much, Mandana. This was amazing. Thank you.